I'm really glad to be here, and I certainly have to start my presentation by congratulating Alexis on a great thesis presentation, a great thesis defense, and a great thesis. It was fun for me to be part of the team that helped him through. We would have monthly Zooms. We only missed a few, I think, in the last few years, and it was just fun to kind of banter and say, "Oh, that's really not a good experiment," or "That's a great experiment," or "Don't listen to Will on this one." Will has a good idea. But it was really a great, something that I've always loved to do is mentor students. And what I'm going to give you today is some snippets of my experiences, both as a trainee and a trainer, and hope that that gives you some insights into, things that you might encounter as we move through your career. I hope this is going to be very interactive. So I will stop and ask you questions. I won't point anyone out, but we'll ask the groups of questions because some of the encounters were kind of fun and a little wacky. I should see if they go through. But thanks everyone for coming. We're online. Don't see anyone from my lab online. No. So let's see. Oh, I can use this for the system move forward.
Second from, it should be third from the top, I believe, is forward.
Oh, I see.
There we go.
Got it. Okay, so I have to thank Will and Chris, and the CAIRIBU community for actually giving me this opportunity and also sponsoring me to come. I've been pushing for 2 years to be here live for Alexis Defense, and so I was glad I was able to do that. So thanks to Will and Chris for that, for allowing that to happen. Just to kind of give you a little bit of background and it'll become relevant as I go through. Yes, I got a PhD at Yale, postdoc at UCSF. I was fortunate enough to work with 2 mentors who are now National Academy members. I think both of them got their membership while I was in the lab, which was really kind of fun. An exciting time for them, the 2 of them. And I've been a faculty member since 1985, probably before any of you were born. In terms of my training experience, I've had quite a few PhD students. I thought I finished my last student about a year ago, but one of Will's former undergrads, Michael Bust, came to me and said, "Hey, I want to change mentors, and I know we've been working together for years, and I finished my PhD as part of my MD PhD program in your lab." I'm like, I thought I finished doing this, but I said, of course, Liv, and she's my last student. I say that now, I really mean it. And she should be finishing in about a year. I've had a number of postdocs, clinical fellows, because I've been in medical school for a while, and TMTC, too many to count, undergrads, medical students, and served on many, many, many thesis committees. I've been very fortunate to be supported for many, many years. I can't say uninterrupted. I think I had a year somewhere where I was in between. But what I want to do is highlight something that you might consider in your careers is to really think that you can do anything. So my very first grant was from actually, my very first grant was from NCI, actually. I'll talk about that in a second. But I have a grant now from NICHD, NIDK, part of some grants as a FOI, aging, I think is our grant will, heart, lung, blood, some DOD, bunch of times. So I think, and even NSF when I was first starting out. So I think what I've learned throughout the years is that if you have an idea and, and reach out to people who can help you develop that into a fundable project, one should never be afraid of what to do. So now let me talk about some experiences I had as a trainee. So my PhD advisor was years old. Who was actually a postdoc here at Madison, worked for Govind Khurana. I want to ask the group if anyone knows what Govind Khurana is famous for and what Dieter was actually part of when he was here in Madison, probably in the late 60s. Khurana, name sound familiar? Okay. genetic code, identifying the triplets for each amino acid. That was work done in Karana's lab here in Madison. He got a Nobel Prize for that, shared with a couple of other people. Dieter was a chemist at the time, came from Germany, who synthesized the triplets and did some of the assays that identified the code. So there's definitely a Madison connection here. He loved Madison, he loved his time here, and certainly was a big part of this amazing discovery back in the 60s here, the practitioner of the code. As a postdoc, I worked with Keith Yamamoto. And what I'm going to kind of explain to you is, you can work from mentors that are completely different approaches, and yet learn from each of them, what you feel would work best for you. Right. And I think that, and that could change over time. It doesn't have to be the same way, but I think I've learned from both of them something very different. When I was in Dieter's lab, he was exploding, he was traveling a lot, he wasn't around much. So I really relied on a big lab. We had a big lab of about 15 people. I relied on a lot of people in the lab for the day-to-day help. In Keith's lab, Keith was there. He was there a lot. We used to joke, we kind of figured out, a couple of us figured out that at night he would go through the lab and peek in your notebook. So we figured that out. And someone was there late and kind of watched him like, oh, he's going to everyone's note. So we would make little jokes, so that the next morning he'd say, I saw what you did. But a very different experience working for someone who was really well established at theater was, always traveling. He loved traveling. He had a lot of connections in Europe. Seemed to be in Europe all the time. And this is before we had internet and Zoom and cell phones. So it was very hard to kind of really get some guidance from him. But when he was here and you wanted to talk to him, he was available. But it was not that frequent. Whereas Keith was there all the time. So what had happened in both labs? Well, as a graduate student, I'm aging myself now, so I'm just gonna have to apologize. I didn't walk to school in the snow and barefoot. So I started my graduate work in 1977. And at the end of my first year, I decided to join Dieter's lab. And I asked a question that probably many of you have asked class: what are the available rotation projects? What are the available thesis projects? And at the time, I was very intrigued by this one publication just came out from this lab, where they used nuclei hand isolated from Xenopus oocytes. So in case you don't know, Xenopus frogs, Xenopus laevis, have like huge, females like huge oocytes, and I will say it's possible, but it is possible to actually hand isolate the nuclei with a dissecting scope with a pipette. And at the time, there were no real transcription systems available in vitro. And there were a few labs at the time that figured out that if you hand isolate these nuclei and gently break them up, you can cover active RNA polymerase III and transcription factors and use that extract to transcribe RNA polymerase III driven genes. Well, it didn't work for RNA polymerase II, but you could at least transcribe cloned 5S RNA, transfer RNA genes in the system. So they published that first paper in '78. I said, Dieter, I'd love to work on this. And he said, transcription is dead. You're not doing that. We're not a transcription lab. Remember, he cracked a genetic code. And so his lab is open. And even to this day, he retired literally, I think 2 years ago, at the age of 89, still had grants, and has always been working on translation, tRNAs. He's really one of the world's leading experts in that field for many, 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 many years. He said, we're a tRNA lab, we're not a transcription lab. So, dilemma. What should I do? What would you guys do? He said, we're not doing this anymore. What should I have done?
You'd like a mentor, get another project?
Get another project, get another mentor. It's another option. Ignore your mentor. Ignore may not be quite the right word, but I kind of did. I mean, I was a second-year grad student, just beginning, or actually in my second year, so I was just beginning in the lab. And I was honest with this leader, okay, I get it. You know, there's all these projects in the lab that have to do with tRNAs and amino acyl tRNA synthetases. You have these projects, you have bacterial projects, you have RNA modification projects, heavy-duty chemistry stuff. I said, let me at least play around a little bit. And so I was honest with him, and I think the pressure in those days was not to quickly finish your PhD, but to do a little bit of exploring. And first thing I did was I cloned another set of tRNA genes. And I found one, shown gene 2 here. These are both tRNA genes that have the same coding sequence, the same tRNA species, tRNA lysine 2 from Drosophila. I found one that worked really well. I convinced Otto, I said, Otto, please, could you isolate some more of these nuclei for me? He said, sure. I found one that worked really well, transcribed tenfold better than another one. So what do? So again, one thing that works and one thing that doesn't. So I have thrown out that bad gene. What would be the first thing you would think of? If you have a system that works, and you have two different plasmids from two different genes that have the same coding sequence. And what I'll tell you is the promoters for these tRNA genes have been known in the last 6 months when I was there. The promoter was within the coding sequence itself. So that gene had the right coding sequence. Right? What do you do? Well, first thing I did is I made another prep of that plasmid. I asked Otto, hey, I need more nuclei because maybe this bad prep that you did. There were times when the frogs, both sides were not so good. So there seemed to be some seasonal variation. So I must have done this experiment 3 or 4 times with 3 different DNA preps, 3 different preps of oocytes, and I had the same outcome. One gene was transcribed really well, one gene was not. Well, since both genes had the same internal sequence, the same coding sequence, I basically took out the flanking sequences of both and showed that they both transcribed well. So what that meant, at least the conclusion I made, was that maybe there's something upstream of the promoter of that gene that is somehow blocking transcription. So I just made a fusion where I swapped the 5' ends. And the swapped 5' end from this gene shut down transcription of the promoter from this tRNA gene. And that was my first publication as a grad student, showing that in fact, for RNA polymerase genes, even though the promoter is contained within the coding sequence of the tRNA, there could be an influence from external sequences. And then Dieter was very happy. And in fact, even though he told me in '78 that they're not a transcription lab, they continue to do transcription stuff, RNA Pol 3 transcription for another 15, 20 years. Isolating transcription factors, doing more genes, blah, blah, blah. So I felt like at least I had an impact on where his lab was headed. And, and that's just what I kind of get to this point. Like, what are the lessons learned? I wasn't combative with my thesis advisor. I didn't say, no, I want to do this, I'm going to do it. I didn't bail and say, okay, I'll do what you say. But I was honest and said, hey, I'd like to give it a shot. Give me a few months to see what I can do. And, I'll start with these 2 genes, see what happens. And, not only did I have a nice thesis in the end, but a contribution to the field, but also contributed to the lab because they continued doing transcription for a while. And some of the postdocs that stayed in the lab, their careers were based on RNA polymerase 3 transcription and output. So a lot of lessons here. And it may same thing may happen to you when you go as a postdoc. You may be on a project, or you may join a lab where everything is pretty well defined, and this is the box you're going to fit in. But if you have an idea based on something they may have done or something that someone else may have done, I think being honest and at least asking for some time to explore, it's never a bad idea. So that was the first lesson I learned. Even though at the time I was totally petrified, like, how could I tell this potential Nobel Prize winner, he never got the Nobel Prize, but National Academy member, that I'm not going to listen to him. I didn't say it like that. That's ultimately what I did.
Hey, Don, a quick question for you in regards to, as you're making the decision, transcription, translation, the boss saying one way and the other. Have you met your better half at this time?
Yes, I was, we weren't married yet, but Helen and I were together. But she was in school too, so she had to just do what you want to do.
Okay, so that's what I was gonna say is, the importance of bouncing ideas off of
Yeah.
important people.
No. So I have to say that I had some really good support from people in the lab. I mean, this guy Otto, who was on that paper, was great to say, hey, I'm busy, but okay, I'll isolate, because he basically had to sit there in front of a dissecting scope and just individual oocytes, he'd pick out the nuclei, and he's great at it. But he was willing to help me out. Of course, he got sick in authorship, and he went on to do other things when he finished, and not at all related to this. So I had support from him, and then later on, other postdocs helped as I went further, and basically dragged them into this transcription field. And then Dieter became excited too, because now it turns out at the time we were doing this, his other projects were starting to stall.
Sure.
And that was a lesson that he kind of also imparted upon me, is that it's not sometimes a good idea to have multiple projects going on as a PI, because some things may stall, and you'll always have a second or third project that might move things forward. So we were basically in the late 70s. The lab was more transcription than it was tRNA processing or anything. They were really interested, and at the time, Dieter was trying to prove that processing of RNA, that RNA was a catalyst. Maybe you may have heard that in your courses, that RNA itself can be a catalyst. And he was trying to prove that. Unfortunately, he was beat by Sid Altman, who was at Yale, floor above us, who was one of the people who showed that with Check and others. So he didn't get the Nobel, but he was really right on the edge. And so that project was stalled. And this took off and actually supported the lab for another 10 years minimum. But yeah, it was, I was very fortunate to have a partner who said, okay, do what you want to do. She was in school, so it didn't matter because she got a job right after, made a lot more money than I did. So for a graduate student, I could linger, although I didn't take that long to finish, because once I had this result, everything just kind of laid out. So I think a lot of lessons here. When I think back on it, I don't, kind of embellishing a little bit. I never said to Dieter, I'm not listening to you. Mm-hmm. I was always very honest with him and said, look, I really want this. Can I give it a shot? And was open to moving on to these other projects if I had to. So let's go to Keith's lab. Different scenario, different dilemma. I was working on a project that had a grant for 6 months in. It was really moving slowly. But I was trying to get it to work. Then Keith said, well, I know this is really not what you're doing, but we would really like to identify a binding site on DNA for the glucocorticoid receptor. No one had identified, the specific sequences that nuclear receptors bind to. One assay that was available was this thing called the footprint assay, which a few labs at Harvard were doing at the time. And what you basically do is you take pure protein, you bind it to DNA, you cut it with DNase kind of lightly, and the region where your protein is bound is going to show up kind of uncut. Well, that's why you call it a footprint. All the DNA around is going to be cut, and where it's protected because it's bound to a protein, it's going to be understood like a footprint. It wasn't my main project. We had an idea. We didn't have really good resolution, but we had an idea that there was a GR binding within like a KB or so of some element from a virus, but we didn't really identify the site. He said, could you do this? Could you at least get the assay working? My job was to get the assay working. Well, the assay worked. And as a result, you can see this. What you're looking at here is lane A. It's a DNA cleavage pattern of this fragment of virus that was known to be regulated by glucocorticoids. And in these lanes B through E, you're adding increasing amounts a purified glucocorticoid receptor. You see this hole here. So identify the binding sites, and these are the binding sites that were within, D20 nucleotides, so you can identify a consensus element for this receptor. This was at the time when there were a number of labs working on it. I think we came out around the same time. Now, back in the day, the group 3, people didn't do co-first author stuff. It just wasn't a thing. So I sacrificed, this is the most important data in this paper, although there's plenty of people who contribute other things. But, nowadays I would be co-first author, but I wasn't, because I wasn't part of the initial finding. So I was second author. So that was kind of the compromise I made, but I contributed to the lab, and it basically gave me some credibility in the field. And this is really what got me started in the glucocorticoid field, was basically this result. And it came out of, once again, the lesson being, sometimes you have to step back, and the advisor really needs help. If the lab needs help, you have to make some sacrifices. Luckily for me, the project that I was stumbling with just got trashed, so I never went back to it. And I continued on this path of identifying binding sites, other viruses for the receptor using the same assay and others. But it was definitely a risk and a sacrifice. Say, put my project on hold. So I was funded for. I had a NIH fellowship or some other fellowship. I put it on hold, but it helped the lab, and ultimately, helped me as well. So this is what I continued. So, once again, two different kind of scenarios. One where I was the initiator of the action, said, I want to do this, and please give me time to do it. The other where my mentor said, hey, we need you to step back and help us out. Both cases, I was fortunate that it got me. So something to keep in mind. And I will definitely give you a couple of pearls of wisdom that I still remember from Keith and Dieter. And this is one of the arguments Dieter gave me when he said, no transcription. He said, look, it really doesn't matter what you do as a PhD student, what the topic of your thesis is, just learn to be a scientist. So forget the transcription thing. I know you like it, but don't worry about it. You can do something else. And even though in the end I did transcription, I think he's right. Because I went from I did work on transcription, but it's polymerase 3, tRNA genes, Drosophila. I went from that, working in Keith's lab on glucocorticoid regulated transcription. So it was still kind of transcription, but it was, a little bit different. And I think I've seen over the many years that this phrase really does bear out. If you become a good scientist, it doesn't really matter what you do as a PhD student. The topic is not relevant. Just do it well, like Alexis has done, and you'll be trained to be a scientist and have the capacity to do other stuff. Something I learned from Keith, sometimes, the hard way, is in lab meetings, he would say, look, you got to read in areas outside of your own, because that's where you get your ideas. Go out of your comfort zone. And he would always push us to read literature on transcription and replication in bacterial systems. He would say, you got to understand the lac operon and lac repressor if you want to understand how glucocorticoids and other nuclear receptors regulate transcription. And he would tell us that, and he would remind us that at lab meetings. He would say, oh, great result. How does that relate to what Patashni showed with lambda repressor in a paper, genes in development, or there was a genes development wasn't even around, PNAS, 9 months ago. Like, okay, good question. We get back to him on that. But I think it's really important to kind of keep in mind that you really needed, and this is before PubMed, before these kind of searches, AI for sure. But still, I think it's even more important now, but it's easier for you to read outside your field. And you'll see some examples of that too. That's a pearl it's really remained with me. And the other thing that I would say about Keith is that he was incredibly slow, but very, very thoughtful and articulate writer. And he would always say, every word should count, every word. You know, it would take us months to get things published. And I have to say, even though Dieter was a good writer, there's a big contrast, because I remember my last meeting with him, like a week before I left to go do my postdoc. He said, "I'll bring down any data you have that you haven't published yet." So I brought down some data. I showed it to him. He goes, "Okay, let's write this paper." I said, "Okay, you know, when do you want to do it? I'll be there next week. You know, I could do it when I'm." "No, let's just, what do you got? You got an hour? Let's just do this." Are you kidding me? You know, so we sat there for an hour and we just whipped out this paper. Now, I'm not saying it was horribly written. Would it win any, you know, literary prizes? Probably not. But that was his style, you know, because he knew that once you leave the lab, it's very, very difficult to finish up a manuscript. And you might encounter this as you move on. And if you don't, you should make sure that when you leave a lab as a postdoc or a grad student, you have to come to some understanding with your mentor. I tell all of my students when they're leaving, my postdocs, I say, look, you know, even if we submitted a manuscript, I said, look, I cannot guarantee the authorship will be the same if you leave now. Not that I want to keep you. You will still be in the paper, but I cannot guarantee you're most likely it will be first author if the work is, you know, mostly yours, but you may not, you may be co-first. But I made it clear at the very beginning. And even at the very beginning, I tell people that. Now, once again, not because I want them to stay longer, but because I want them to say, hey, look, you know, if we can't finish it, I'll do everything I can to get this. And it's up to you. If you want to go to a higher impact journal and the risk is higher, that the author list may change. So I had a student who we published like a genes and development paper, something like that. And we sent it in, an EMBA journal paper. So it's pretty reasonably high impact. Sent it in, and it tons of revisions. The student was leaving. I said, look, you know, so-and-so is going to have to do all of these revisions. So he became second author. Now, nowadays, he would have been co-first, but not first listed. You know, and I had, you know, other scenarios where I had someone who collected all this data, never analyzed it, never wrote the manuscript. Someone came later, did all the analysis, wrote the manuscript. And I had to say to the person who would now be gone 3 years, look, we're going to submit it. You're going to be called first because you collected all the data. The person who was first author collected zero data. But he did all the analysis, and it was a clinical project, so it required a lot of analysis. So I think you have to be mindful. It's a good conversation to have. You know, one more scenario that was kind of a little different was I had two, a postdoc and a student, write a review paper. And I said, they both wanted to write this review. I said, okay, how about authorship? I said, what do you want to do about this? And they said, well, we'll leave it up to you. And they both took a crack at writing this and they put it together. And I said, I really don't know how to decide. I said, look, the only thing I can do, I went back to the very first draft, each of their parts of the manuscript, and I word counted 100 words. I didn't tell them that beforehand, because otherwise there would have been a little bit of extraneous stuff in there. And I counted words. And I said, you're both go first, but so-and-so, Lexi's going to be first because she had 1,500 words. And I'm sorry, Kim, but you had 1,488 words. I have no other way to make that decision. You know, and they both said, okay, because we gave you that, it was your decision, we gave it up to you. They had come to me and said, no, Lexi really wants to be first. Then as a mentor, I could have said, okay, then Lexi's going to do a little bit more of the busy work for this. But they were both so equally involved, I had no other choice. So, you know, it's always a good idea to kind of have some conversations about, you know, what the expectations are without being kind of onerous about it. But I think those are 3 examples of kind of what I did. Now, let's go on to being a mentor. And there's so many, I'm sure you go on to courses and you've seen, you know, podcasts about the transition from a mentee to a mentor. So I will give you some personal reflections on some of my most wacky examples, maybe some lessons learned. And that's been been in Pittsburgh for years. So it's been a lot of years doing this. So hopefully the examples I give you will provide some insight. So first thing I'll tell you about is kind of reflective of what I mentioned here about how many different agencies have funded me. My first grant as a new assistant professor was on glucocorticoid regulation of transcription of retroviruses. Well, that wasn't going so well because we published one other paper 2 years after I left Keith's lab, identifying binding sites for the glucocorticoid receptor and other retrovirus, but it wasn't a big deal because there's really not a lot of biology that was associated with that. Still not known what that means. But we took a 180 turn and went to study how retroviruses or oncogenes within retroviruses affected the receptor. And that was, once again, based on an observation of one student who was working on nuclear trafficking of the receptor. And we were still trying to figure out how to examine the regulation of retroviruses like glucocorticoids. But he noticed something odd. He said, there's something odd about these cells that have these retroviruses and oncogenes, that the trafficking of the receptor is different. So, and this is the paper that was named that kind of showed that. So we made a complete 180 to study kind of the opposite end. It was based on one observation, looking at a cell that had this retrovirus oncogene and one that didn't, and just paying attention to what he was, your data, like the receptor is behaving differently in these cells. So I think that's another lesson I think I'll get to in a second. And what that led to is more work on now the trafficking of the receptor. So we went from now looking at how oncogenes affect trafficking to how other factors might affect trafficking of this receptor in the nucleus. And this allowed us to kind of move into the neurodegeneration field because what we found was that there were certain mutants of this receptor that had a tendency to aggregate. You're looking at immunofluorescence micrographs, black and white. Nuclei are kind of tell them, but yeah, the DAPIs here. But this one mutant of the receptor tended to aggregate nucleus. But you can overcome those aggregations if you co-express a particular chaperone protein. So this is the same mutant when you co-express the chaperone. So remember the J field is not important. At that point in time, it's like, okay, it's great. There's a weird mutant receptor that aggregates, so we can eliminate a nuclear aggregate with the chaperone. Once again, I go back to what Keith said about looking outside your field. I said, okay, what other nuclear aggregates are more physiologically or pathologically relevant than just this wacky mutant receptor? Well, at the time, there was a lot of interest in the formation of nuclear aggregates by polyQ expanded proteins like Huntington protein. This is the taxon-1, spiral taxon. So there was, at this point, it became known that there were nuclear aggregates that contributed to neurodegenerative diseases. This is back in 1998. So I contacted, after reading some papers by Scobie, I said, hey, I know you're working on nuclear aggregates, taxon-1, this disease. I think you can eliminate those aggregates in animals or in cells if you overexpress the chaperone. And that's what happened. So we jumped into neurodegeneration because of our interest in trafficking of the glucocorticoid receptor, because of a result that was maybe not that interesting, because it was a very rare mutant receptor that we just happened to show form aggregates, but we jumped into a field where nuclear aggregates were much more relevant to pathology. So that enabled me to kind of spend a little bit of time working on chaperones and aggregation and neurodegenerative diseases and how we got some of the funding in NINDS. So once again, what do you think I've learned from all of that? Back to what Keith said. Read outside the literature. Read outside your comfort zone. Don't be afraid to do that. If you get a result, you trust the result, but think beyond the interpretation of that one piece of data. What can I, how can I take that data, the data set, whatever, and apply it maybe a more meaningful translational or pathological or whatever state. And so I think, once again, it's an important thing to keep in mind that, and I'll go back to what Dieter said, because as long as you're trained to be a scientist, you can jump fields. Now, it's not always easy. I will definitely make sure I would emphasize that. It's not always easy to be the dumbest one in the room. When I gave a talk on some of our chaperone nuclear aggregation stuff at a neurodegeneration meeting, I really felt like, that wasn't like I felt like I was dumbest one in the room. But I had some collaborators, people at Pittsburgh who were more experts in neurodegeneration who didn't mind me asking stupid questions, where I felt a little more comfortable asking stupid questions. So it may be difficult at times to do that, but reach out to people you feel comfortable with, give you some guidance. Because even if you read the literature as I did, you don't always get all of the insights about what's relevant, what's not relevant, what models you can use, what models you can't use, things like that. But certainly I've learned over the years that, hey, just step out of your comfort zone, and maybe it's because my upbringing, I've just never, it doesn't bother me to embarrass myself. That's kind of my mantra. Like, so what? I'll wake up tomorrow morning and I'll embarrass myself again. But if you have people that are willing to help you, and if you're willing to do the work to kind of catch up, you can really do a lot of things that you become now the expert in. And I remember going to a meeting 5 years later where I was considered the expert, chaperones and neurodegeneration, to even have a patent for a drug, which of course went nowhere. But still, 5 years later, now I'm the expert, which is fine. It took a little while to get there, but you can see how the transition happened. Pay attention to your data, and then always ask yourself, what does this mean outside of my little scope of work? Okay, so more little reflections on students, trainees in my lab. So I had a really talented MD PhD student in the lab who had published a paper. He had written a grant on this neurodegeneration and some other model, oxidative stress, doesn't matter. He got, I forget, like a 15% or 18% from NANDS. Didn't get his proposal funded. He was really upset. And then, I was having trouble getting my grant renewed. And I said, okay, Ramal, we're going to change your thesis project. We're going to work on neurodevelopment and glucocorticoids. And he looked at me like, kind of looked at me like this. So what do you know about neurodevelopment? I said, really nothing, but I have a really good feeling about this project. I said this to him. I have a really good feeling about this. And he just looked like this. So I want you to respond. What would you do if you were in this case? What would you ask yourself? What would you ask me? What would you do? Because there are plenty of things that went on beyond this conversation. But this is the initial conversation, and this was his initial look. If your mentor said, I got a good feeling about this, and you're already a third year, and you're going to change your project. What? Go for it?
Say boss.
Whatever you say, boss.
Big painful thumbs up, though.
I know. Oh, God. Well, I mean, would Ramal have been, would it have been appropriate for Ramal to ask me, like, why do you have this good feeling?
Yes. Who's gonna help us if neither of us know about it?
Yeah, what is this good feeling? What are you talking about? What is this good feeling? How you know? And so it was upon me to say, elaborate even further. I didn't have a ton of preliminary data. We had nothing. I mean, I hadn't done anything in neurodevelopment. But I said, hey, Mama, first of all, we have neurodevelopmental people, downstairs, people that I know who are really like, I know glucocorticoids are important in neurodevelopment. I just don't know how. But I know glucocorticoids, I know GR. And I have somebody who can help it. But once again, I was the dumbest one in the room when it came to starting this project. But he was real. I'll tell you, he did not give me the thumbs up. He did not. He was really upset about this because he was his third year of an MD PhD program and a graduate program. And he's like, I already got a paper. I already have some data. I didn't get my fellowship. You didn't get your grant renewed. Why are we doing this? I said, I'm going back to my roots. I'm dragging you with me on this journey. But but to be honest, over the next few weeks, I did try to elaborate more with him. I read more. I talked to him about what I was reading. I said, okay, let's do this. And he reluctantly, I think, aboard. What resulted was first author of paper. Yes. And a really, really cool story that was very new to people because we identified a non-genomic effect of glucocorticoids in stem cell, in clustered stem cells, or what I would call coupled stem cells. So we started growing stem cells. They're kind of like organoids, but not really organoids. This is 2011. They weren't really organoids. You can think of them as organoids, but they're coupled. The cells are coupled. We knew that. And we knew that there were techniques, and we had people who were helping us with this called PrEP, rapid recovery after photo bleaching. So you load every cell in this cluster, in spheroid, organoid dye, and a dye that can move through gap junctions. You bleach one cell, and then you wait, single bleach, and then you wait to see how long it takes for the dye to come back in. The dye can move through gap junctions. So we had a lot of controls. And we found out that, in fact, glucocorticoids regulate gap junction communication. And the important thing about that was this tracing here, which is probably one of the coolest data pieces that ever come out of my lab. This is a trace of individual cells in these nucleus neurospheres. These are individual calcium traces, individual cells. And you can see that they're pretty coupled, right? And we did some statistical analysis of that when there's no hormone treatment. When you treat with hormone, it completely get uncoupled, which fit with the data here when you analyzed the recovery in the presence or absence of glucocorticoids. The other cool thing about this project was that my first thought, I said, Mama, it's been shown in pancreatic beta cells or something that glucocorticoids regulate gap junction protein expression, RNA expression. That must be what's going on. So let's do a Western blot. You do a Western blot of one of the gap junction proteins cells, plus or minus hormone expression. What's going on? So he came in this blot. He said, in the RNA, which there's no change, and said, there's no change in the protein level either. So your idea that glucocorticoids regulate gap junction communication to regulating expression of these gap junction proteins is this particular one. It's completely wrong. And what did I say? What do you think I said? Look at the data. What did I say? You're right, Rama. You're wrong, Rama. Missing something, Rama? Check out this little smudge. The smudge was brighter. What does that smudge represent? Phosphorylated forms, that protein. Because I had done the reading and found out that these proteins are regulated, their activity is regulated by the state of phosphorylation. And you can see that in the Western because the phosphoforms run a little bit higher. And Ramal showed me this blot. He was really bummed. I was like, yay! He said, what are you talking about? I said, look at that smudge. He said, what about it? I said, it's brighter. There's more phosphorylation of this protein. And so that's what led him to do other studies, identify the kinase, identify the sites. And in fact, that's the model. That's what we showed is that non-genomically glucocorticoid receptor influences MAPK, ERK1 and 2, the influence of phosphorylation of this gap junction protein. It shuts down its activity. And he had all the controls and that's what So, lessons. Sometimes
thumbs up is okay. But it's okay to also ask your mentor to, give a little more context here. Okay, I need more than it's a good feeling, If you want me to do some more reading, fine, but I need to know what you know. Why you have a good feeling. That's the first thing. Second thing was look at your data and have someone else look at your data. Ma looked at his data because he actually had this data first. He had the gap junction question first before we had the frat worked out. Have someone else look at it and look into, once again, do the reading to say, how is this protein regulated at the level of its expression or the level of its phosphorylation? And how do people measure the phosphorylation status? They do that by looking at these smudges that have higher glycan weight mobility in FDS gel. So Mal's now pretty happy with me, invited me to his wedding. And I still talk to him. He's a super nice guy. He's an academic physician at UCLA. He's still doing neurology. He got another paper on kind of epilepsy related study we did with collaboration with some other people while he was still at my lab. So it turned out really great, both of us, and it was fun to kind of, see this whole process emerging. Now, I would have to admit, once again, to be all honest, in all honesty, I was playing around with him a little bit when I said I have a good feeling. I did have a good feeling, but I still needed to do a little bit more work. So I was playing around with him a little bit, but I think in the end, it was a great progression to kind of really work out a really cool mechanism. And that kept me funded for a while from NICHD. And so we get glucocorticoids and neural stem cells. It's less than there. So now I'm going to bring back some prostate and Madison stuff from a paper that I did with Will that was done by Teresa, you may all know, stock here. And this is a paper she published with us in JPC. And it was based on this notion that cyclooxygenases, COX-2, in particular, might be regulating steroidogenesis, or the metabolism or the production of estrogen ligands, or even testosterone metabolites. And so we had a really good once again, having a good collaborator, we had a great collaborator, Rich Hawkes, great mass spec. And this just shows you kind of the control mass spec, where we isolated from these cells, focused on testosterone, hydrotestosterone, and these various metabolites. If you spike the cells with this treated or deuterated testosterone, you can see its metabolism of DHT and these other guys. If you spike with DHT, you see the metabolism of DHT for these other guys. Fine. So Teresa looked at cells where she depleted COX-2 because we had some evidence that COX-2 impacted estrogen signaling. And look what happened. So T to DHT, that step in the metabolism of T is not affected. But the conversion to these other metabolites, which are up here somewhere, don't worry what they are. You can see one, this is androstenedione, I think. 1 and 2 really severely impaired. And when you spike with DHT, you can see the same thing. DHT does not get converted to these other metabolites. So we said, great, COX-2 is regulating some of the enzymes, if not many of them, involved in the interconversion of dihydrotestosterone, these DR-beta ligands, because we were focusing on estrogen such as beta, blah, blah, blah. So this is great. So the Teresa did an RNA-seq, COX-2 knockout cells, normal cells, did the GO analysis, and looked for the pathways with steroidogenesis. One of the pathways she found, the answer was no. So now we are in a quandary. We think that COX-2 is regulating many genes involved in steroidogenesis. We did the RNA-seq. I think it was IPA or whatever, pathway analysis. We didn't find steroidogenesis at all. So what do we do? Now this was back in 2016. Pathways we didn't have AI then, right? So you can't use AI. And the pathways, they're more elaborate now. There's more than one pathway tool. But But what kind of discussion do you think Teresa and I had? Is our hypothesis completely wrong? Is the pathway analysis completely wrong? Should we look in to see, like, how does this go for this IPA? I think we use that one. How do they actually define their pathways? That's something you may encounter, because what I'm learning now is if you use different pathway tools, you sometimes get different results. But before that, as a Teresa, just I'm going to just hand select. Let's do old school. Let's just do RT-PCR, all of the enzyme RNAs for all these enzymes of these genes. Let's see what happens. You knock down COX-2. And that's what this table shows. You don't need to see the details. Bottom line is nearly every the mRNAs for nearly every gene in this ureagenesis pathway were affected by COX-2. It was not picked up by the pathway analysis, but it's picked up by digital RT-PCRs. So I think we confirmed that our hypothesis was correct. So lesson there is trust your data. I think trust your data, and if one tool, and once again, even AI, I have to tell you, I use Claude a lot. Definitely found Claude makes mistakes. There's absolutely no doubt on mine. I'm not saying a lot, but it does make mistakes. And I can also tell you that Claude gets very defensive when you call him or they out on mistakes. When it happened, I basically had two age-dependent changes of two different genes, one with this one with that way. And I showed Claude the data, and I asked Claude for an interpretation. And Claude said, "Oh, these genes are, co-regulated during aging in the same way." I wrote, "Claude, did you look at the graph? They're in the opposite way." "Oh, sorry. No, you're right." Then I asked, "Well, why did you make that mistake?" And then it got really defensive. "Well, these two genes, are in a complex, and they probably should be regulated the same way. And, to the data I had at hand." I'm like, "But I gave you a figure." So anyway, what was I getting back to? Yeah, trust your data. And if you think a tool is not, and if you have other data supporting a certain hypothesis, and the tool that you're using, whether it's AI or whatever, is giving you something that you don't expect, it might be right. But, try another tool or learn what that tool is using to generate those pathways in this case. So, I don't know how much time I have, but
A couple
more minutes. I was going to talk about this too, Will, but since Lexi did a great job with this, I won't do that. But I will say, I will do it quick. So we started this collaboration with Will looking at inhibition of mitochondrial activity, complex 1. And this is a kind of a cool story because it came from an undergrad who was here, Keegan, who was basically treating cells with rotenone. We use the same stromal cells that Lexus uses, but we grow them in galactose to really make them struggle. And what Keegan said is, "Oh, these cells are dying, floating. When I add rotenone and galactose, they're floating off, they're dying." And I said, "Well, are they really dying? I mean, yes, they're floating, they don't look good, but are they really dying?" It turns out, when this is Laura's work, did a lot of work, don't worry about the details, the cells weren't dying. They were floating off clusters, they weren't dying, they were still alive, but they were becoming resistant to anoicus. Like, I remember this word from whenever, but I'm like, what the heck is anoicus? Right? It's a form of cell death that occurs when cells that are normally attached become detached. Right? And it's really most important in metastasis when cancer cells leave their site and they move into the circulation, that they become anoicus resistant. So metastatic cells that are moving in the bloodstream are anoicus resistant, because normally if they're adenocarcinomas, they're derived from epithelial cells that are attached, they come off, they would die, but they become anoicus resistant. So we said, so once again, back to the literature, what the heck is anoicus resistance? Well, what does that have to do with BPH? Found one paper, which in fact wasn't a very good paper, but it linked anoicus resistance to fibrosis, which I know certainly people here are very interested in the role of fibrosis, BPH. And this paper mentioned this passage of this one gene that seemed to be a player. Didn't have really good data for it. But I'll spare you the details, but, you know, Laura and a lot of people large group of people found that actually this protein, which is a transcription termination factor, plays a role in mitochondrial function, organs resistance, cell attachment, and this is the basis for R01 she received. So once again, trust your data. Get your data. You know, as an undergrad, I didn't expect Keegan to know about ficus. I didn't know about it either. But at least, you know, she showed me the data and I said, you know, I've seen a lot of dead cells and that are floating, and you know, we've all had that. These don't really look dead to me. So, you know, you gotta go. Let me tell 2 quick stories because I think the lesson that I think is important. But let me tell 2 quick stories about networking, because I'm sure you've heard about it. Got a network, and that's certainly true. But it's not always easy. And I'll give you 2 examples. My first example, 1979, I got invited to this very prestigious conference, Gordon Conference. And I told my mentor, Dieter, he said, great. I'll call Mark Natasny, who's a professor at Harvard at the time. He's chairing a session. I'll ask him to give you some time to talk in the session. This is very commonly done at these conferences. I said, okay, great, Dieter. So excited. We get to the conference. I had this whole memorized. Dr. Natasny, blah, 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 blah. Dieter called you and said, and he goes, who are you? I never got a phone call from Dieter. I'm like, now what? Well, what do? Well, I just sucked it up. And the day that I was supposed to, that Bethany's session was, I reminded him, you know, he said I could have a few minutes. So he goes, okay, fine. You have 5 minutes to talk. So it became an important part of my career because it gave me so much confidence that there were 2 or 3 Nobel Prize winners in the front row, all these people from Harvard, you know, Stanford, you name it. And it was a real booster, but I was really ready to strangle Dieter. I tried his cell phone and I was like, Dieter, what the F are you doing? There's no cell phones, nobody to get a hold of him. But, you know, sometimes you just have to take chances and have the confidence in yourself that no matter what, and this was a real blindside. I got really blindsided here, you know, because I was like, what, 24, 25, you know, and this guy was really famous and he's a Dieter, I was going to strangle him, but I went through. Last thing. Once again, networking, it's not so easy. It's a very important lesson here. It's at a conference, a student tells me, well, you can't really see it, but I'll tell you, the student said, oh, I was looking at a poster, she goes, oh, this is our work, and you know, it really clearly shows that this work by this guy DeFranco is completely wrong. This is true, this is true story. And I said, you know, congratulations, I really like your data, great work. And she went, oh, who are you? I said, I'm Donald DeFranco. So, listen here. Read the badge. I mean, this is kind of a funny lesson, but it did really happen. And I felt so bad for the student. I really said, that's okay, don't worry about it. I said, there's other interpretations of all of our data, but I think you did the right job. But I said, maybe you should try this instead, but read the badge. So, yeah, read the badge. So anyway, thanks for all sticking around.